Okay, good morning, ladies and gentlemen. Thank you for being here. It's my pleasure to introduce Dr. Joseph Chen here to give a seminar as a bioengineering faculty candidate. And he comes to us from UC Berkeley, where he's currently a postdoctoral fellow and actually studying cancer, and he's going to tell us a lot more about that. He did his bachelor's and master's at the Mississippi State University, so much closer to us than from Berkeley. And his PhD actually at Vanderbilt. And so he's actually has family in the area as well, I think Tennessee or so we're very happy to have him here, and let's give him a good strong welcome.
All right. Well, thanks for the introduction, and thanks for having me. I'm excited to share with you all a little bit of the work that I've been doing at Berkeley and tell you a little bit about the future directions of my research. So when I began my career in science, I was motivated by the goal of developing new drugs for treatment of disease, and that led me to study the etiology of various diseases. And so I studied primarily fibrotic diseases and cancer, and that accounts for the vast majority of diseases that affect Americans. For example, if you look at some common or some statistics on fibrotic diseases, you'll find that fibrotic diseases such as pulmonary fibrosis or cardiovascular disease, so valve fibrosis or infarct regions, they account for over 45% of all deaths in the United States. And then if you look at cancer, as we know, cancer is the second leading cause of death in the US and accounts for about 22% of the deaths. And as I began to study these diseases, I was really kind of struck by this common thread that runs through these diseases. There's a process that we see in cancer and we also see in fibrosis. So what is that common thread? So it's mesenchymal transitions. And in mesenchymal transitions in disease, you see in fibrosis, EMT inducers will lead to the activation of the resident cell population. And that causes the cell to secrete more ECM proteins and become more contractile. And that leads to cell death, calcification, things that drive fibrotic disease. In cancer, and metastasis specifically, cells have been observed to undergo a type of mesenchymal shift, which enables them to become more aggressive in their motility and invasion, and also makes them more resistant to therapy. So we see here that mesenchymal transitions in disease, they're actually critical in both the onset of disease and disease progression. So what do we know about mesenchymal transitions? We know actually a pretty good bit, and we know most of this from the developmental biology space. And kind of the general description is that the epithelial monolayer of embryos, when they undergo gastrulation, they lose that epithelial phenotype, and they acquire more mesenchymal phenotypes, which enables them to migrate away from the monolayer of cells to form more complex tissue structures. And actually, there's been a lot of research in the EMT field in the last 20 years. It was first described by Elizabeth Hayes in the 1980s. And actually, the first description of it is just a developmental program that enables stationary epithelial cells to gain the ability to migrate as single cells. So this seems fairly simple. But over the years, people have then more recently described this process as a complex and diverse process that's activated by intrinsic and extrinsic factors that are finely regulated temporally and spatially. So I think that's a sentence that just says it's way more complicated than we initially thought. And actually, targeting of this mesenchymal transition has been really challenging as well. As I mentioned, most of the insights we've gained have been through developmental studies. And through those studies, there's still been a lack of robust biomarkers. And actually, you'll see that over the last 10 years and even beyond this, the research activity of this field is just skyrocketing. and with the goal of finding more biomarkers. And as I mentioned earlier, in these developmental contexts, the cells are in a monolayer, and then they become more complex. But in disease, these cells are actually situated in very, very complex environments already. So many bioengineers, and myself included, have noted that the EMT in disease occurs in a very, very complex microenvironment, and it's subject to a variety of biomolecular and biophysical cues. So deeper investigations are needed to uncover new mechanistic insights in this process in hopes of identifying new targets for pharmacological intervention. And more recently, in the last 8 to 10 years, people have really started to unravel the mechanobiological contributions of this process. So in valve disease and valve fibrosis, the cell is promoted to become a myofibroblastic-like cell through applied forces such as mechanical strain or tissue stiffness, as well as signaling factors such as TGF-beta. A recent study by Wei was able to look at the intercellular tension distributed through monolayer cells and show that very finely regulates the MRTFA accumulation in the nucleus. And in the cancer space, you can see that there is this kind of interplay between the biomolecular signaling and the biophysical cues, looking at matrix stiffness and TGF-beta signaling, showing there's some synergy in promoting this metastatic behavior of these cancer cells. And to kind of move forward a little bit in glioblastoma, this is the context, the microenvironment that the glioblastoma cell finds itself in. And there's tons of niches that I won't go through at this time, but every cell, in this environment is subject to biomolecular and biophysical cues. And so this is the situation that we find ourselves in, and this is the complexity of the context of the disease that we find ourselves in. So how do we really approach this? How do we study this to really dissect the mechanism in a physiologically relevant system? So this is where I hope to make a contribution by taking a mechanobiological approach to the system. So we just envision a cell in this complex microenvironment, subject to stresses, strains, the tissue level, applied level, and from its neighboring cells. So how should we tackle this problem? And so obviously, we have molecular biology tools to kind of dissect the protein states, the genetic states of the cells. But we can use engineered platforms to recapitulate some of these microenvironmental cues by making, I guess in the brain specifically, HA platforms that can give you that biochemical signal from the cell binding to the hyaluronic acid, but also give you that stiffness cue. We can also use microfluidic devices to recreate some of the geometries that we find in these systems to try and elucidate the effect of them. We can also use genetic tools, and this is more of tools to really understand the mechanotransductive effects of the cells. So we can inducibly, in a graded fashion, induce activity and modulate the actin and microtubule architecture to really see how these external forces are transmitted within the cell. And lastly, we can use biophysical tools such as AFM and TFM, and also FRET to really quantify the forces that are involved in these processes. And so this is my approach in investigating this question. And I'm going to switch gears a little bit and tell you a little bit about the work that we've been doing at Berkeley, utilizing these approaches in trying to understand this process. Okay, so a little bit of background about glioblastoma. Glioblastoma, or GBM, is the most lethal malignant primary brain tumor, and it represents around 50% of all primary brain gliomas. The median survival time is a dismal 1.5 years, and recurrence is inevitable. And even though there's been tons of research on this disease, the prognosis still remains poor. So why is this? So glioblastoma is a very, very aggressively infiltrative, invasive cancer. And you can see in these micrographs the borders of these tumors. They're very diffuse and they're actively spreading. And what happens clinically is most of the time you remove that primary tumor and you can't get rid of all the tumor material. And there's residual tumor cells that will then go and form secondary tumors and lead to patient death. So the first challenge is that this cancer is very, very aggressive and infiltrative. But another reason it's so difficult to treat is that current conventional treatments, such as radiotherapy and chemotherapy, they actually drive mesenchymal transitions. So they can be effective initially, but then what we observe is that eventually these tumors will recur, and they'll recur in a more aggressive state. So in this state, they're more resistant to therapy, and they display enhanced invasiveness. So one great approach to tackling this question is really focusing on this mesenchymal transition process. So regarding mesenchymal transitions, what do we know? And this was just a review paper I looked up, and I just thought it was kind of funny because the authors literally just threw everything together and put it in a cell, and they were like, this is mesenchymal transitions. When we look at this, it's hard to figure out what to target. So what is the approach that I'm taking? How do we target these mesenchymal transitions? So first off, I'm interested in looking at the glioblastoma stem cell. And so this is a certain population within the tumor mass that has some stem populations and are what's thought to drive secondary tumor formation and migration away from the primary tumor. What our approaches can be to, one, disrupt pro-invasive mesenchymal changes. And one story I'll get to would be focused on the actin dysregulation that occurs in GBM. Another strategy is to prevent or reverse mesenchymal transition. And within the diagram that I showed in the previous slide, there is something that we can focus on, and these are the transcriptional repressors of E-cadherin. And so during a mesenchymal transition process, cadherin switching is the initial thing that occurs. And so if we focus in on understanding how these mechanisms are regulated, perhaps we can stop mesenchymal transition before it starts. So for the projects that I'm going to talk to you about today, and I also just mentioned, I'm going to walk you through a story talking about cytoskeletal alterations in GBM, and specifically ones that are heightened in the mesenchymal subtype. And then I'll move to a second story focusing on the mesenchymal transitions that occur and with the efforts of halting it at the cadherin switch. So the major objectives of these projects are to elucidate the underlying mechanisms of actin regulation in mesenchymal GBM, and then to also interrogate the mesenchymal transition pathways to identify key promoters in heightened cell invasiveness. And I'll talk to you, and I'll bring you guys through this by talking about these two projects. So project one is looking at this protein called limb kinase, which is an actin cytoskeletal regulator. And our goal was to investigate what its role is in GBM stem cell invasion. And the second project will be focused on cadherin 11 and mesenchymal transformation to examine the role of this particular cadherin in driving mesenchymal shifting. So we'll start by talking about limb kinase. So this work was motivated from some previous work in the lab, which was interested in looking at mechanotransduction of cancer stem cells. And some previous work in the lab showed that when you took a cancer cell and you put it on substrates of varying stiffness, what you really would get is very spread, very motile cells on stiff substrates and pretty poorly spread cells with low motility on soft substrates. When we took this particular type of cells, the cancer stem cells, and we put them on these gels of varying stiffness, what we saw is that irregardless of the stiffness, the cell spreading, the cell motility, they were all the same. So we used some of our inducible constructs to manipulate Rho A signaling because Rho A can regulate contractility and contractility can regulate mechanosensing. And what we found is that when we constitutively activate Rho A, we restore this kind of stiffness-dependent response where the cells were balled up on the low stiffnesses but very spread on the high stiffnesses. And this had effects in invasion. And when we took these constitutively active Rho A cells, the cells couldn't invade to collagen. And even in transwells, the migration was reduced. And so when we proceeded to animal studies, we confirmed that this finding, this mechanism, or this phenomenon was observed in vivo as well. But we really can't target Rho A easily, so we were interested in understanding what downstream effectors may be important in regulating cancer stem cell invasion specifically. And so we kind of went to the literature and we found this protein Lin kinase. And we found that Lin kinase is situated at this node which is regulating actin polymerization and depolymerization. So very quickly, Lin kinase is situated above cofilin, and it phosphorylates cofilin to inactivate it. And when Lin kinase is not there and cofilin is not phosphorylated, then it's actively cleaving the actin cytoskeleton. And the cell uses these mechanisms to coordinate the cytoskeleton to facilitate efficient motility. Well, we're really intrigued about Lin kinase also because of its role in 3D invasion. And as you know, cells in vivo, they don't really, whatever we see on a 2D plane doesn't necessarily transfer over to in vivo. So 3D invasion is markedly different from 2D invasion. So we found this paper kind of detailing Lin kinase's role in affecting 3D invasion in breast cancer. And then we also found that Lin kinase 1 and 2 have prognostic value in that, when they are downregulated, you have overall better survival. So we decided to focus in on Lin kinase 1 and 2, namely because it was downstream of Rho A, and it had all these effects that seemed to make it relevant for in vivo migration. And so we started by probing the Cancer Genome Atlas, which is this database that has genetic, epigenetic, protein level, RNA level changes across 33 human cancers. And so we were able to see the % alterations of these cases in GBM. And so we compiled a list of motility, actin-based motility genes, and we saw that Lin kinase 1 was dysregulated, or upregulated rather, in 21% of GBM. With Lin kinase 2 has a much lower alteration at 5%. But when we look at the alteration on the Kaplan-Meier survival chart, what we see is that the patients that have an alteration or upregulation in Lin kinase have poor survival. And when we compared these genes with glioblastoma versus low-grade glioblastoma or low-grade glioma, what we saw is that Lin kinase 1 was significantly upregulated in comparison. So this gave us enough evidence to proceed to investigate Lin kinase in our system. And so we tried to do some loss of function studies. And so we use PLKO vectors to knock down Lin kinase 1, Lin kinase 2, or the combination of Lin kinase 1 and 2. What we saw was interesting in that cofilin phosphorylation, which is the main target of Lin kinase, it wasn't affected until you did a double knockdown, which was new information to us. When we did SIM imaging looking at the cytoskeleton, what you can see is really clear. In the non-targeting and the single knockdowns, you still get the kind of standard cytoskeletal architecture that you would expect in a cell. But the double knockdowns, what we see is very, an accumulation of these puncta of actin or G-actin throughout the cell body, with only these peripheral fibers intact. And we made some videos to really investigate the dynamics of this process. What we see is that in the non-targeting cell, you get this classic lamellipodial extension with retrograde flow. And when you look at the double knockdown, what you see is really a random mess of actin polymerization and depolymerization. So the actin turnover in the Lin kinase knockdown cells is constant. And so our question was, what's the functional consequence of this? And so we naturally went to just 2D motility, kind of the most standard motility assay to quantify cell speed. And what we saw was somewhat surprising is that we do see some cells that display kind of aberrant behavior, but through the population, we weren't really able to observe any significant changes. Excuse me. So we got some kind of surprising results. We got this huge result at the actin level, but then it didn't really manifest itself in 2D. And so that's when we moved forward with some more engineered approaches and more biomimetic systems. And we started by putting these cells in our 3D HA-RGD spheroid assay. And so this is an HA material that we've methacrylated and cross-linked with DTT. And we basically embed a spheroid of these GBM cells in and monitor them over the course of 14 days. And what you see is pretty clear. In the non-targeting, we get pretty kind of consistent invasion outside of the sphere. But in the double knockdowns, we see very, very little invasion. And that's quantified and shown here. So with this biomimetic platform, we're able to then tease out the differences between the non-targeting and lymph kinase double knockdown cells. So we then proceeded to try and kind of segment the contributions of this data, so what the cell may be experiencing in a tight 3D confined gel versus a 2D surface. So we moved to investigate more confined types of invasion assays. So the easiest one is the transwell assay, and we basically had 8 micron and 3 micron pore sizes. And we saw that we see these differences in these double knockdowns in 8 micron and 3 micron pores. And the difference is more pronounced in the smaller pore sizes. So this was exciting to kind of confirm the HA data. But we really wanted to be able to observe more finely what's happening in this confinement space. Because in these HA materials, the cells have to migrate through a confined matrix. So we made these engineered microlines of different widths, and we put these non-targeting lymph kinase cells on these surfaces, and we tried to quantify cell speed. And what you'll notice is that in the more confined spaces, so in the wider spaces, you're not seeing any differences, presumably because there's a lot of 2D motility going on as well. But when you narrow down the channel width, you then start to tease out the differences between the non-targeting and the lymph kinase cells. We then looked at chemotaxis because within the transwell assay, we do create a chemotactic gradient, and we see that we get significant dysregulation in chemotaxis in the lymph kinase double knockdown cells. So here we've really shown through our in vitro data with these engineering platforms that lymph kinase 2 regulates confined migration, 1D migration, and chemotaxis. So we took this data and moved forward to the cancer stem cell population. And we created these double knockdowns, and we also did the same imaging, and we did the transwell assays. And we were able to show, for the most part, that the lymph kinase double knockdown produced the same phenotype in these cancer stem cells, which is important because the cancer stem cell population, as I alluded to previously, is the population that we want to target ultimately in an in vivo setting. So that's what we did. We moved forward with an animal experiment looking at 2 time points, 7-week time point to basically compare the non-targeting and double knockdown at the same time, and then an endpoint. So then we extended this experiment till the endpoint of the animal to characterize tumor growth, survival, and do some staining. And what we saw was very, very clear and really exciting. So in the double knockdown cells, when we injected them into these animals at 7 weeks, what you see is that the signal that we get from the tumors is much, much smaller, and that leads to a much lower quantitative metric in bioluminescence. And when we monitor the survival and the growth of the tumor, we see that the tumor growth is delayed, and also we get about a 30% increase in overall survival in these animals. When we looked at the tumors and H&E staining and also in immunostaining, what we see is that the tumors are much smaller. And so the tumor occupancy that we quantified also demonstrates that or describes that. And when we look closer at the tumor sections, what we see kind of confirms our hypothesis that in the non-targeting, you get much more invasion at these margins, at these tumor margins, but in the double knockdowns, we have much clearer borders. So our conclusion with this is that the lymph kinase knockdown tumors are not able to infiltrate. So they're kind of kept into this tumor mass, and so therefore they display reduced tumor size and dysregulated tumor development. So in this project, what we really showed is that lymph kinase 1 and 2 is upregulated in GBM, and it's actually most highly regulated, upregulated in the mesenchymal subtype. And the knockdown of lymph kinase leads to a hyperactive cofilin, which leads to this this constant turnover. It reduces cell invasion by slowing confined migration, by preventing proper cell polarization, and disrupting cell chemotaxis and directional cell migration. And we also show that this is true and relevant in vivo, and that lymph kinase suppression reduces in vivo tumor spread and extends survival. So this is exciting because it's the first paper to show that lymph kinase inhibition can work in vivo, or has some in vivo benefit. So we're excited to move forward with this, and this paper currently is in review at Cancer Research. So to bring you back to this slide, I want to tell you another story about our efforts to prevent or reverse mesenchymal transition. And I'll move forward toward my project focusing on cadherin 11. So before I get too far into the role of cadherin 11, we need to do a little bit of background to understand the subtypes in GBM. So GBM right now, and I guess more recently, this is from 2010, but more recently they've taken out the neural subtype. But essentially, you can think of this disease as a spectrum of subtypes, with the proneural being associated with better survival and less invasion, and the mesenchymal being associated with worse survival and more invasion. And during the GBM disease process evolution, and during recurrent GBM, mesenchymal shift is often observed. And how does this happen? So as I mentioned earlier, there are conventional treatments such as radiotherapy and chemotherapy that lead to mesenchymal transition. And the mechanisms that people suspect that this is happening by is through TGF-beta signaling. So in patients that are post-radiation, they see that there's an enhanced SMAD2/3 phosphorylation, which is indicative of active TGF-beta signaling. And this activates mesenchymal programs such as Snell and Twist. And in a study looking at anti-angiogenic drugs, what they showed is that the progressive hypoxia leads to an accumulation of macrophages, which increases the release of TGF-beta. So in these conventional treatment modes, what you're essentially creating in the microenvironment is an increase of TGF-beta, which leads to increases of TGF-beta signaling, which is a potent inducer of EMP, and so reports suggest that it can also promote PMP. And so proneural to mesenchymal transition is kind of a term that we and a few others have started using, but it's within this transition process, there are these cadherin switches that are proposed to occur. And this is true in many, many cancers. I mean, many cancers of epithelial origin, there is a switch from E-cadherin to N-cadherin at the very onset of a metastatic transition. And so what happens is these kind of epithelial sheets will downregulate E-cadherin and upregulate N-cadherin, and these cadherins can intimately affect the cytoskeletal state and control motility through OGTPA signaling. So one thing that hasn't really been kind of fleshed out in GBM, because it's not an epithelial cancer, is this cadherin switching occur in this cancer, and does it play a role in cell motility? And so our approach to this was to see if we can identify a cadherin switching process in GBM by mining the cancer genome atlas. And then we want to characterize these proneural and mesenchymal subtype cancer stem cells. From there, we want to develop an in vitro disease model to determine the role of cadherin 11 in GBM, GSC invasion, and then hopefully uncover new mechanisms for cadherin signaling in GBM. So similar to the lymph kinase project, we started by mining the cancer genome atlas. And so we started by seeing these kind of classic cadherins in the EMT process, cadherin 1 and cadherin 2. And what we saw was that there were no changes between the subtypes in both of these cadherins. And this is actually a really interesting place of research, and many people have been looking at N-cadherin in GBM, and there's been this controversy in the field, oh, it's not here, or it is here, and there's been a lot of confusion. because people have just focused on these specifically since they've been established as EMT cadherin proteins within the disease. But we didn't see anything different, and that led us to kind of explore more cadherins. And what we found was that this specific cadherin, cadherin 11, was significantly different between the proneural subtype and the mesenchymal subtype. So this is the first evidence that there could be this other cadherin switch that may be involved in GBM. So we noted that CAD11 is significantly upregulated in the mesenchymal subtype specifically. So what do we know about cadherin 11? We know that it contributes to increased invasion in various cancers, but specifically in the breast cancer field, they noted that this cadherin was only found in the most invasive breast cancer cell lines. And CAD11 is also associated with the poorest prognosis cancers. And the kind of interesting thing and exciting thing is it provides, it's a type 2 cadherin, so it provides a somewhat unique domain for pharmacological targeting. What's also interesting is cadherin 11 has been found in fibrosis as well, in valvular calcification, and also rheumatoid arthritis. So we wanted to proceed and see maybe this cadherin protein is playing a significant role in GBM. And so we, with our collaborators at MD Anderson, we acquired a panel of proneural and mesenchymal subtype cancer stem cells, and we evaluated the expression of these proteins across those lines. What we found consistently is that within the proneural subtype, what you get is a stronger expression in N-cadherin. And that's not actually surprising because N-cadherin is actually neural cadherin, so it's ubiquitously expressed throughout the brain. But what's interesting is that in the mesenchymal subtypes, you have very little N-cadherin, but you have kind of a dramatic increase in cadherin 11 expression. And this is associated with increased phosphostat-3 and total STAT3, which are mesenchymal markers. When we did our staining, we do see that in the proneural subtypes, we get strong staining at the cell-cell junctions for cadherin 2, or N-cadherin. And in the mesenchymal subtype, we do see some cadherin 11 at the cell junctions. When we look at CAD11 in the proneurals, I don't show this data, but you really don't see anything with respect to CAD11 and vice versa. So we move toward quantifying the kind of invasive potential of these subtypes, because it's one thing to say that the mesenchymal cells have higher cadherin 11, but maybe what if they're not more invasive? Then there's not much you can really say about cadherin 11 in invasion. And I'll also say that we did this experiment in kind of your classic 2D plastic experiments, and we did a lot of experiments and got really no difference between these cells. We moved toward an HA platform, a 2D HA material, which provides these CD44 binding sites for the cells. And then we all of a sudden started seeing these dramatic differences between the proneural and mesenchymal subtypes. So these are all done on HA-RGD gels, except for this figure, and I'll talk about it in a second. So this is essentially 2D motility, looking at cell speed and persistence on 2D HA-RGD gels. And what we see is a dramatic increase in the mesenchymal subtype in their ability to migrate and navigate HA matrices. The projected cell area shows that they're much more spread on these HA materials. And this is an adhesion assay. And we did this to try to see how these subtypes interact with the HA matrix. And so what we see is that in the mesenchymal subtypes, the cells are much more adherent to the material and presumably much more able to spread and interact and move. So we also moved into the 3D HA platform, and we saw the same thing with the mesenchymal subtype, that they're much more invasive through this 3D matrix. And so you can see in these images from day 1 and day 14, the sphere looks essentially the same, but in the mesenchymal subtype, you can see the invasion at the periphery, at the edges of these spheroids. So here we demonstrated that CAD11 is upregulated in mesenchymal, and the mesenchymals are also much more invasive. So the question is, if we can force a proneural cell to become more mesenchymal, and by creating a disease model where we induce mesenchymal transition, that gives us a window to really evaluate mechanisms that may be driving these changes. So we looked at TGF-beta as the inducer of mesenchymal transition, and we essentially treat these cells for 7 days and then look at the mRNA changes. What we saw was really exciting because it confirmed that we are able to take these proneural cells and force them down this mesenchymal transition pathway. And so we see that for cadherin 11 and CD44, the mRNA levels are significantly increased with TGF-beta incubation. And what's interesting also here is that in the EMT markers, we looked at snail and slug and ZEB1, and only in ZEB1 do you get, you get an increase in mRNA level expression. In the mesenchymal subtype, it's a different story. You get really no change in CAT11 and a slight decrease in CD44, but you get these massive increases in snail and slug, but no change in ZEB1. And this is actually an area that I'd like to pursue further, understanding the different roles of these transcription repressors in this type of cadherin switch. When we look at the protein level, we see that CAT11 is significantly increased in the proneural subtype as well as in CD44. We also see increases in phospho-STAT3, which is a mesenchymal marker showing that these cells are undergoing this mesenchymal shift. What you see in the mesenchymal is very minimal change for TGF-beta, which is not surprising because they have very high levels of cadherin 11 and CD44 to begin with, and perhaps they're responding to TGF-beta in different ways than the proneural would be. So then we looked at adhesion, motility, and invasion. So kind of the metrics that we evaluated previously to see if we take these proneural cells with TGF-beta, will they then functionally be different? And we do see that. With TGF-beta treatment, we have a pretty significant increase in cell adhesion to the matrix, to the comparable levels of the mesenchymal subtype. And we see that with TGF-beta, we get increases in cell speed and also in 3D invasion. So then we move forward to some loss of function studies. And this is kind of where we're at in this project. So this is somewhat still preliminary, but we basically used CRISPR to knock out cadherin 11 in the system and did some kind of preliminary tests to see what the knockdown was doing. So if you look at the proneural mesenchymal spheroid invasion, you can see that the mesenchymal cells are spreading much more rapidly than the proneural. But what if we took the mesenchymal subtype and we knocked out CAT11? So in this case, we see kind of a behavior that kind of looks like the proneural subtype, and we see a dramatic reduction in that space. When we look at TGF-beta treatment of proneural cells, we were wondering if it could reduce the pro-migratory effects of TGF-beta. And what we see is that it indeed does. So the current state of this project is as follows. So we identified a novel in-cadherin to cadherin 11 switch in GBM, leading to upregulated CAT11 in the mesenchymal subtype. And we show that mesenchymal glioblastoma stem cells are more invasive than the proneural ones on HA platforms, specifically in vitro. And then we show that mesenchymal transition can enhance the way the proneural cells can interact with the HA matrix, and that CRISPR knockout of cadherin 11 can robustly reduce GSC cell invasiveness. And so some of the next steps in this project, which is ongoing and which is research that I'd like to bring with me, is investigating the role of CAT11 in regulating this mesenchymal shift. And we're actually testing this right now where we take the knockouts, and if we have, the cell does not have an ability to make cadherin 11, does it actually undergo a mesenchymal transition, or does that inhibition of the cadherin switch affect its ability to go through the mesenchymal transition process? And so we'll look at qPCR of EMT and use TGF-beta treatment of the knockouts. We can look at a lot of protein things as well. And so I'm also doing experiments right now to determine the role of CAT11 in facilitating cell invasion. So the interactions that it has with CD44, there's evidence that cadherin expression and CD44 expression are linked. And then looking at downstream effectors. So if you have a mesenchymal cell that has Cat11, there's an intracellular complex that links with the cytoskeleton and links with certain signaling pathways that are very interesting. And so I'm creating these mutants essentially to test those hypotheses. And another thing I'm doing right now is assaying some of the biophysical changes that are associated with mesenchymal transitions. That can help, that has diagnostic implications, but also can help us better understand how the cytoskeletal state is being regulated when the cells are undergoing this mesenchymal shift. Okay, so I take you back to this slide because I'll briefly touch on some of the proposed experiments or work that I'd like to do at my next step. And so I think as an engineer, I think we all like to use tools, and I think the ability of the questions you can ask are heavily dependent on the tools that you have. And so taking a mechanobiological approach, I'm a mechanobiologist, and there's a lot of things that we aren't able to ask if we don't have the appropriate tools. And so using the engineered platforms, the genetic tools, and biophysical tools in conjunction with the conventional molecular biology tools can give me a lot of potential questions to ask in various disease contexts. And I'm not aware of who has kind of received a previous version of my talk detailing specifics of the proposed projects, but I'll just briefly go through some of the ways in which I'll use these approaches to ask new questions. So I'm really interested in neurodegeneration. I'm really interested in astrocyte activation. And it's been more recently reported that there's this astroglial mesenchymal transition that occurs after injury. And the extent of injury, the extent of activation determines the patient outcome. So if we can understand the mechanisms that are driving this post-injury process in, for instance, traumatic brain injury, then perhaps we can control the kind of recovery response and improve prognosis. And a second part is this, as you've already seen, which is a continuation of these glioblastoma-focused projects. So I just wanted to show you this diagram of some of the approaches that I'll have to basically study neurodegeneration and cancer. So within the kind of TBI-focused neurodegeneration research, it's going to involve some outside-in tunable biomaterials. So in that project, there's this idea of taking tumor slices and doing some AFM to understand the mechanics of what changes at the tissue level, and trying to recreate those materials to provide these external cues for the cells, to then study how the cell interacts with these changing external environments. And so within the GBM space, as I'm kind of alluded to, is this inside-out cytoskeletal mechanobiology. So downstream of cadherin signaling, there's tons of proteins that are intimately involved in actin regulation. So how do those mechanisms play out, and how does that intrinsically affect the cell and how it's transducing signals from the outside? So I'm very interested in taking an inside-out approach to cytoskeletal mechanobiology. And then at the end, what kind of brings everything together is this NGS, next-gen sequencing approach. And I think in our field, it's rapidly changing. And I think we as researchers need to actively adjust and adapt to those changes as well. And as these technologies become more readily available, we need to adjust and to apply those into our research questions. And one thing I'm really kind of intrigued by, and this may be somewhere further down the road, is epigenetic sequencing in the context of mechanobiology. And there's some preliminary reports that say, there are these epigenetic changes that occur within the cell as they encounter different mechanical environments that kind of functions as a memory for the cell. Where if you take a cell on a stiff gel and you put it on a soft gel and compare it to a cell that's always been on a soft gel, they're fundamentally different. And that could be due to some of these epigenetic changes. So taking these astrocyte activation questions and the GBM questions, eventually bringing them into the epigenetic space, I think will yield new results. So really, I just want to put this up briefly to kind of talk about these kind of two main projects that I brought up. This is just really an overview, and I'll be meeting with some of you later, and we can discuss some of these in detail. But project 1 is focused on the astrocyte activation studies, and then project 2 is looking at cadherin regulation in GBM. So I promised Herman I would keep this around 40 minutes. So I'm going to move to the acknowledgement slides and just thank Sanjay Kumar, my PI, my mentor at Berkeley, and for all his mentorship, his guidance, and his help in my postdoc. I'd like to thank my collaborators, Joe McCarty at MD Anderson, and my colleagues, Elena and Carmela, and my undergrads, Rena, she's at the Buck Institute now, and then Leslie, Sharon Gokul, Frank, are all undergrads that are working with me currently. And I'd also like to thank my funding sources, the NIH and NCI. So with that, I'll stop here and take any questions.
I was wondering if you could explain why you chose hyaluronic acid as your platform for your biomimetic scaffold.
Yeah, for sure. So, we're studying brain cancer, so everything's in the context of the brain. And the brain's a very unique tissue. It's a very unique ECM, and it's comprised primarily of hyaluronic acid. And so that was the main motivation to using HA platforms, because we want to understand how these cells will navigate the brain matrix. And so we're using HA to kind of be to mimic that.
And then when you've done your knockdown studies and so forth for the signaling, some of them were in the HA platform from what I could tell. Have you looked at different stiffnesses then of that to see how that environmental cue links into what you've seen?
Exactly. I mean, I think that's a really great question, and that is one of the things we are proposing to study. So we did have a previous paper where we did modulate stiffness of HA, and we can do that by changing the percent HA pretty easily. And we, in that paper we did see that there was a stiffness response, and that promoted increases in motility and invasion. But for this study right now, we've kept everything more physiologic at the moment. And if we want to focus more on this, we ask the stiffness question in this context, we can do that as well.
Other questions?
So I have a question. You mentioned that the application of your work will be brain cancer or brain tumor. This may be very good for short vision, but do you have any plan for long vision? Can you investigate other cancer, other type of cancer?
Beyond brain cancer?
Yeah.
Yeah, so I think the beauty of investigating mesenchymal transitions is that, in the statistics that I've shown, the relevance of this process extends to all those disease contexts. And actually EMT and this whole cadherin switching question, it's been pretty well studied in many other cancers. But in brain cancer specifically, it's been pretty confusing because it doesn't follow that standard paradigm. So I could envision taking some of these questions and continuing to ask them in those other cancers. But another point to bring up is these tools that I've been able to utilize. I think it brings utility to many other disease questions. I think that's what's exciting about the field of mechanobiology is even though it's kind of understood that these mechanical inputs play a role, it's still not as well studied, and I think primarily because we don't have the tools to study it. And so as these kind of tools come online, then we can kind of go back in some of these disease contexts and ask these mechanobiological questions, which I think have a significant impact in disease progression.
So another question, one of the main strengths of BE at the University of Global is we focus on translational research. Do you see some of your work can be can be translational, because we have here many programs internal in Global cover translational research like Holter, Excite, and recently we have innovation grant from Vice President of Research. So,
Yeah, so, I think that's one thing I'm really excited about. I think by training, I'm a basic scientist, right? So I'm really kind of dissecting mechanism of disease processes and things of that nature. And I think the most translational type of stuff that I've done to this point is really, identified drug targets that then led to collaborations with people that, are, doing research on drug delivery, finding ways to get the drug to where it needs to go, and things like that. But, I think I'm very open to explore more translational or more clinically focused projects. But at this point, as a basic scientist, I'm here understanding how things are working, and I think that does have a good place to interface with clinicians.
I had a question. So, targeting these proteins in other neurodegenerative diseases is probably possible because the vasculature is potentially still intact and things can be delivered. But in a tumor like the brain tumors, how would we deliver drugs to target these proteins in the cells that are potentially far away from vessels, near collapsed vessels, and that we won't get there to change their
and I think
what that challenge.
Yeah, I think treating anything in the brain is challenging with the blood-brain barrier even, and in the parenchymal space is very tight. And I think that's really a drug delivery question that I think there are some faculty here at Louisville that specifically look at that using smart materials or nanoparticles or some other approach to where it needs to go. And so I think that's a very relevant question, a very interesting and important question that we need to answer, for some of these therapies to really have an effect. But right now, I think for me, it'd be better to kind of talk to or collaborate with an expert in drug delivery to try to see if we can kind of interface our expertise to come up with a solution for that problem.
Any other questions?